Okay, good morning. Thank you so much for joining our part 3 series of our Patient Empowerment Educational Series. This series is presented by My Style Matters, Tia Tomlin, who is the coordinator, founder of that organization, and Cancer Awareness Network, of which I am the coordinator and founder of our organization. We're two nonprofit organizations who touch the lives of so many cancer patients dealing with this dreadly disease. And not only do we work with the patients, but we actually work with their families. And our goal is to bring some sense of light to their journey and to help them as they navigate the different experiences of dealing with cancer. And because cancer is a whole gamut of things come into play, we decided, since we could not service our people, our patients in person because of the pandemic, we decided to come up with this platform, which is a Zoom presentation. So we're actually in part 3. We did part 1 in May, part 2 in June, and now we're close to the end of part 3. We have one more presentation after this, which will be held next week. And so if you're registered for next week's conference, please be mindful that you'll get a notice because of the conflict we had with Dr. Williams. We're going to start maybe 30 minutes earlier. So I'm just excited about being able to bring this presentation this morning. And I also want to say that those that are registered, you will be able to receive a gift. We do a drawing. And so the gift for this series is coming from Hello Cure, which is an organization down in South Alabama who do these beautiful gift baskets. I also want to give a shout out and thanks to our sponsor, Amgen, who has helped us to put these platforms together. And so the way things will go today, we'll have our presenter, Dr. McDaniels, will speak, and then we will, as she's speaking, please be mindful, everyone will be muted. But in your chat box, mine is located at the bottom of my screen, you can always ask questions as the person is talking. And so at the end of the presentation, we will allow times for Q&A. And so with that, I'm going to introduce our speaker today, Dr. Joy A.G. McDaniels. She's a PhD, and she's a postdoctoral fellow in the lab of Dr. I'm not going to pronounce that name right, G. something Melanzo at the University of Texas, MD Anderson Cancer Center in Houston, Texas. Dr. McDaniel is a native of Birmingham, Alabama. She received her BS in biology from Spelman College, followed by a master's in biology and PhD in biotechnology, science, and engineering from the University of Alabama in Huntsville. Dr. McDaniel research is focused on understanding the underlying molecular mechanisms that drive metastasis in aggressive forms of breast cancer. Her dissertation research led to the discovery of a role of the transcription factor STAT3 as a regulator of invasion in triple-negative breast cancers. As a postdoctoral fellow, Dr. McDaniel utilized mouse models of breast cancer and genomic technologies to understand how mutation in tumor suppression of TP53 contributes to the onset and metastatic progression of triple-negative breast cancer. She is an advocate for increasing diversity in biomedical research. Dr. McDaniel is very passionate about increasing community awareness about how breast cancer disproportionately affect Black women. And so I actually been knowing Dr. McDaniel for a while. She's been able to help me with my DOD presentations because, you know, those scientific words gets too long to pronounce sometimes, and I have to call her up. And so I'm just so glad that God has allowed people like her in my life. And I'm so grateful that she's going to present to us today. And so with that, Dr. McDaniel, it's on you. Take it away.
Okay. Well, thank you, Ms. Loretta, for having me today. And thank you to Ms. Tumlin for also having me and sponsoring this event. So what I'm going to do now, I'm going to switch over to share my screen, if that's possible. It says that the screen sharing is disabled. Let's try to get this fixed.
Dr. Joy, I'm going to make you the host so you can share your slides.
Okay, perfect. Thank you.
Okay.
Okay. All right.
Perfect.
Okay. So my topic is a little bit different from what the flyer suggested. As Ms. Herring told you, I am a breast cancer researcher. Breast cancer is something that is very near and dear to me. So I wanted to take a different spin on what you may be getting in some of your educational series, and just kind of give you a picture of what are some of the current things that are being done to understand the biology of aggressive breast cancers, such as triple negative breast cancers. And a lot of focus is on triple negative breast cancer because we know that this is a type of cancer that disproportionately affects minority women, particularly African American women, at alarming rates. So I'm going to first tell you a little bit just about genomics. We're just going to have some basic biology talks just so you can follow the jargon and terminology. And then we'll talk a little bit more about cancer. Everybody here knows what cancer is, so we'll probably breeze through that. And then we'll get to how studying your DNA or the genome has allowed us to understand key concepts about breast cancer. But what are those unanswered questions? And that's where I'll share with you a little bit about my current research and how I am using mice to understand the biology of aggressive triple negative breast cancer. So inside of your body, you have tons of cells. Every organ in your body, it has a function. And at the baseline of all of your organs is a collection of cells. Inside of your cells contains your DNA. So your DNA, it is basically your blueprint for what makes you you. It's the reason your hair color is the way it is. It's the reason why your eyes are the color they are. It is also what discerns different ethnicities as well. So everything about us is all encoded in our DNA. So the entire output of all of your DNA, all of the DNA in your body is actually what we call a genome. So at the heart of cancer biology right now, genomics is paving the way for helping us to understand why some tumors respond to treatments and why others don't. And so the key to understanding the genome is critically important because it allows us to understand disease. And one thing we should know about the genome is that it was actually sequenced about 20 years ago. So this was a major feat. So this was a project that started in 1990 and it lasted about 13 years. And the entire goal of the project was to read all of the DNA in every cell in the body. And this is so we can know where all of the genes are and determine what their sequences are. Now, why do we care about that? If you understand where all of these genes are, then if there's a change to your DNA, like a mutation, then it begins to point toward why do we have disease onset. So back then in 2003, when the Human Genome Project was completed, the total cost of this was $2.7 billion. Now, in 2020, the sequencing cost, it has drastically decreased. Now you can get a genome sequence for $1,000. So you may see different ads on TV like 23andMe and different areas that will tell you, oh, send us a cheek swab and we'll tell you all about your DNA. Companies like that are built off of the basis of understanding the human genome. And so now we can take your genetic information and tell you more things about whether or not you're susceptible to disease or not.
Hey, Dr. Joy, I don't mean to interrupt. I don't know if you have your phone on as well as your computer, but we're getting some feedback. Sounds like two things are on.
Okay, can you guys hear me clearer now?
Yep, that's a whole lot better.
Okay, sorry about
Perfect. Thank you.
So, with the Human Genome Project, we were able to understand where all the genes are. We can do this for a much cheaper rate now. And because it is cheaper, this has allowed us to really push forward in the biology of understanding disease. So just a very key biological concept. So I mentioned DNA. So your DNA is in all of your cells. It's your blueprint. It's what makes you you. It consists of letters that go together in different combinations. And basically, they're going to tell every cell in your body to make what we call proteins. These proteins in your cells, they all have specific functions. That's what makes all of your organs function the way that they do. However, you can have changes to your DNA, such as mutations. So when a mutation occurs, there is a sequence change. And sometimes these sequence changes, they can change the function of the proteins. And we're going to talk a lot about mutations because at the heart of studying cancer, it is critical to understand how mutations drive cancer. You can have extra copies of genes. You can have chemical changes that occur. And then you can also have changes that occur at intermediate steps that prevent your proteins from even being made. And then you can also study proteins and where they bind to the DNA. So all of these things can all be measured through sequencing technology by reading the DNA. So you may ask, how do we read the DNA? So in genomics, some of you guys may have heard about sequencing and getting people's genetic blueprint to understand disease. At the heart of that is what we call a sequencer. Now, this may look no more to you than just a computer. Technically, it's a very high-powered computer, and the cool thing about this computer is that it actually has a camera inside of it that allows us to take a picture of your DNA. So very quickly, what we can do, say if a patient comes into the clinic and they provide a sample, say if a biopsy is taken, we can extract DNA from the biopsy. We can take that DNA and put it on what we call a slide or a flow cell. It attaches to this slide, and then we're going to be able to read all of the sequences of the DNA. And what happens when you read all of these sequences, we can take a picture. So what happens, you're taking a picture of all of your DNA, so your entire genome at one time. So to make this easier to understand, inside of that computer, I said there's a camera, there's a microscope and a camera. So here, this is what the inside of the machine looks like. You have the slide that contains your DNA, and then you have a laser, and then you have an imager. And so what happens, you're going to take a picture of all of this DNA. And these cameras, they're so sensitive, they can detect single molecules of DNA, so they can identify fragments of DNA which are about 20,000 times smaller than a single strand of hair. So we can capture a lot of information at one time. So you may hear people talking about big data, big data. We are in the age of big data. And that's really true, because now we can sequence cancer genomes. We can sequence the genomes of patients that have cardiovascular disease. And understanding this information is really revolutionizing the way that we understand disease. And this is just another example of what the data looks like when we're taking a picture. So each of these different colors on the pictures represent one of the letters that make up your DNA. As I said, you have 4 different letters, T, C, G, and A. So the camera is going to take a picture, and the color reflects what sequence they've identified on the sample. So why do we care about this? Sequencing has really sped up how we understand biology in general. Here, we're just looking at, this is what we call a karyotype. It's basically a way of looking at all of your chromosomes at one time. So when you think about different diseases such as Down syndrome, one of the ways that we diagnose that is by looking at, do you have different copies of your chromosomes? So you look at the entire genome in one setting and say, oh, I can identify a change. So actually, this is an example of a normal karyotype, but I'm using this as an example because we're able to take this entire picture and read all of the DNA that is located on each of your chromosomes. So you're asking, okay, you've shown me something highly technical. Don't worry about all the details and aspects of how the sequencers work, but what you do care about, what you should care about, is that this gives us the ability to read all of our DNA and all of our cells, and then we can detect what is different that may predict why diseases occur. And it's also going to be helpful for how we can navigate therapeutic treatments, which is very important in the context of treating cancer patients. So now that we've kind of got just some of the basic genomics terms down and just understanding what is DNA, how do we read and measure DNA, now let's take a step back and let's just talk about cancer. And I'm pretty sure all of you guys have an understanding of what cancer is. So as I told you, at the heart of all of your organs in your body, it's a collection of cells. All of those cells have special functions. So the cells in our body, they make copies of ourselves. This is how you keep normal, happy, healthy cells growing. This process is what we call cell division. As these cells make copies of themselves, they're making copies of the DNA. So it's very important that when we make copies of our DNA, that it's accurate. Now, when it's not accurate, it can be inaccurate because of maybe a DNA mutation. When it's not accurate, our cell can trigger special mechanisms that will tell the cell to kill itself. And this is what we call apoptosis. So basically, the cell commits suicide because it does not have the correct DNA sequence. And really, this is a protective measure for you, because without these repair mechanisms for the cell to go and die, sometimes the cells can escape the triggers to kill itself, and then the cells that have the bad copies of DNA, they begin to grow. And what happens, they ignore all of the signals in the cell that tells it to stop growing. So that's why we call cancer uncontrolled cell growth, because cancer doesn't care about any of the signals. Whatever the changes are in the DNA, it's giving the cells an advantage to keep growing. And that's what leads to cancer and leads to different forms of cancer. So now that we've gotten that out the way, we'll talk a little bit more about those changes that happen in your cell, because that's what's at the heart of studying cancer. Now, one thing to note is that cancer is genetic. It is all about your genetics. It's all about what's in your DNA. So some people have a genetic predisposition. So when you think about people that get cancers and they say, oh, it runs in my family, there may be people that have hereditary forms of breast cancer, meaning that every cell in your body inherited a mutation. You're born with these mutations. So instead of having the correct sequence, your mother or your father may have passed down an initial mutation. And because you have this first mutation in all of the cells in your body, as your cells continue to make copies of one another and grow, the setting is set for a second mutation to form, or maybe even a third mutation. But people that have genetic predispositions or have inherited a cancer-causing mutation, they are more likely to develop more mutations in their cells, and they are likely going to develop cancer. So there are syndromic types of cancers, such as Li-Fraumeni, where the patients are born with a particular mutation, and very early in life, these people begin to have different types of cancers. Sometimes they're breast cancers, sometimes they're bone cancers, but throughout the duration of their life, they have multiple forms of cancer because their cells are already primed to form a cancer. However, these hereditary cancers are only 5 to 10% of all cancers that are diagnosed. The majority of cancers are what we call sporadic cancers. So what is a sporadic cancer? All of the different cells in your body, they can get a mutation, and the mutations are random. And what happens here, you have normal cells that are dividing, and sometimes as they divide, they pick up that mutation, and then they know now, oh, I've got a mutation, I've got an advantage. Maybe your body is not telling the cell to kill itself, and then you end up with a second mutation. And this leads to cancer as well. But the key difference here with sporadic cancers is that all of your cells in the body do not have the mutation. You did not inherit it from your mother or your father. Anything that you inherit from your parents is going to be in all of the cells of your body. So with sporadic cancer, you may get a mutation that's in the breast tissue, but you don't see this mutation in maybe your lung or your liver. So that's how we end up with all of these different types of cancers, because the cancers originate from the cell type where the mutation is formed. So just as we have breast cancer, that mutation occurred likely in a cell that was in the breast. And in general, we have about 2 million new cases of any kind of cancer a year and about 600,000 deaths. So the key focus in research right now is really trying to understand these sporadic cancers, the ones that are non-hereditary. We seem to have a good hold on some forms of hereditary cancer, which I'm sure people may have some questions about that toward the end of the talk, and we can come back to that. But for the remainder of the talk, I'm going to focus on these sporadic cancers where you just randomly have a cell that gets a mutation and it drives the cancer formation. So like I said, the majority of cancers are sporadic. And what causes the mutations to happen? So sometimes there are certain risk factors that kind of primes our cells to allow a mutation to happen. So some of these risk factors can be cigarette smoking, being overweight, alcoholism, exposure to UV radiation, poor diet, sometimes infections, or even physical activity. However, this also highlights the fact that we can also do some things on our own to protect ourselves from developing cancer. One, by choosing not to smoke, lowering your intake of alcohol, choosing to eat healthy foods, because your DNA, it responds to everything that you're doing. Your body is a function of your environment. So reducing stress, all of these things can keep your DNA happy and hopefully not trigger random mutations in your cell. So I told you at the beginning a little bit about genomics and how we're trying to understand the DNA at the DNA level, why cancers form, and how can this inform treatment. So it can be very important because we can sequence the genomes from healthy individuals. So getting your genome sequence is not just for people that have cancer. You can be perfectly healthy and get your genome sequence. In fact, we encourage that because that helps us to understand what's normal in the general population. And then we also want to sequence cancer patients. And this is where we really learn about what's driving the cancer. So this helps us to understand methods for early diagnosis. What about treatment? What are we seeing in the DNA that might guide what's the appropriate treatment? And then you can also monitor if a treatment is working. So if you see some type of changes, that suggests that the cancer is coming back, or if the cancer is spreading. By sequencing the DNA at all of these different stages before and after treatment, it gives us a clear glimpse of how the patient is responding. So now, one of the areas that has really been revolutionized by understanding the genome and how changes to your DNA lead to cancer is actually breast cancer. So breast cancer is very prevalent in our society, and some cancers we understand very, very well. And one thing before we dive a little bit into the stats about breast cancer is just understanding how the mammary duct is structured inside of the breast. Because remember, I told you all of your cells, they have a function, they do something. And inside of the mammary duct, you have two different types of cells. So if you look at these yellow cells, these cells are what we call the luminal cells. The main thing you need to know about these cells is that they're important for milk secretion, and they typically express estrogen receptor, which we know can drive certain forms of breast cancer, estrogen receptor positive breast cancers. But then you have the second cell type, this outer lining of the breast duct. These are the basal cells. These cells actually perform the contracting mechanisms in the breast to help propel the milk to the breast duct. And these cells, they don't have estrogen receptor, but we don't know what causes the types of cancers that form from these cells. And usually these are triple negative breast cancers. So how do we understand what's expressed in these different cell types right here? It's because of the sequencing technology that I explained to you earlier. So one of the key areas that we're interested in understanding is, well, what is driving triple negative breast cancer? Since we know that these cells function differently from the luminal cells and it doesn't express the same types of factors, it's very critical to understanding. So let's dive a little bit deeper. So I gave you the overview of the duct, and let's just backtrack a little bit and discuss what we do really understand and what we understand very well. So in breast cancer, in your breast cells, you have, in those luminal cells, you have receptors that are on the surface. So these receptors, they're just proteins that have the ability to bind other proteins, typically hormones. So when people say they have hormone receptor positive breast cancer, these are people whose breast cells either express estrogen receptor, the HER2 receptor, or progesterone receptor. Now, why do we care about that? We know where these hormones are binding on the cell, and what happens Under normal conditions, these can actually drive the normal growth of breast tissue. But sometimes you can have mutations that are going to trigger the prolonged signaling, and that's what leads to tumor formation. And that's what leads to breast cancer formation that is responsive to hormones. So by sequencing breast tumors, and this is one of the first major cancer studies that we really got some true information from, we learned that by the expression of these cells, we can target these by knowing if a patient expresses estrogen receptor or HER2 receptor. We can target these, and we have seen much success in the treatment of breast cancer in those patients that express these hormone receptors. And actually, breast cancer, this is a model for studying other types of cancers. Also in prostate cancers, we know that the receptor androgen can also be targeted and be a very effective therapy. So this is what happens when you are dealing with hormone receptor positive breast cancers. But in triple negative breast cancer, you don't have these different receptors. So on the left, you have the hormone receptor positive, you have the receptors, good treatment. On the right here, we have triple negative breast cancer cells. You can see the cell is bare. There are no receptors. There are likely receptors there, but we don't know what these receptors are and which ones are important for promoting the growth of these types of breast cancers. So this makes it very difficult for us to treat triple negative breast cancer. So a little bit about it is that we know that the only way that we can treat triple negative is through chemotherapy. So basically, you're just throwing different drugs at the cell and you're taking a shot in the dark and hoping that it's going to kill the cell. But it's not targeting anything specifically on the surface. So usually the chemotherapies that we're using to treat triple negative cancer, they are toxic enough to at least slow the growth or the progression of triple negative. But there's still a very critical need to understand the biology of this, because we can improve patient survival and outcome if we had something a little bit more direct and targeted. So that gets me into telling you a little bit about the aggressive behavior of triple negative breast cancer. Normally, triple negative breast cancer is diagnosed in women and it's already progressed. It's already at late stages or a high histological grade, and usually has a poor prognosis. In simple terms, these are usually not early breast tumors when they're diagnosed. They have progressed far along. A lot of times, these triple negative tumors, they may be progressed enough where the metastasis has already began to form, or it's already began to spread to other areas of the body. So these are highly aggressive breast cancers. In women that have BRCA1 or BRCA mutations, they sometimes, most often, will develop triple negative breast cancer. Now with BRCA, these are women that have a hereditary or genetic predisposition to cancer. They have inherited a BRCA mutation, and this has increased their risk for developing breast cancer. And then other characteristics of triple negative, which I've mentioned earlier, is that it is disproportionately affecting minority women, particularly African American and Hispanic women. And usually these women are diagnosed at earlier ages. Traditionally, these are women in their mid to late 30s that are being diagnosed. So a lot of times when you think of any type of cancer, you think it's a disease for older people, but it's not. Triple negative is hitting young women at alarming rates. So not only do we need to understand the biology of it, we need to develop better treatments so that we can increase survivorship for women that are diagnosed with triple negative. And like I said before, right now the treatment is just chemotherapy, but we want to improve upon that. So as I told you, a lot of times triple negative is aggressive, it can spread very quickly. So we call this metastasis. So when your cancer begins in the breast, it can spread to the brain, the bones, the lungs, or the liver. So this is what we really want to stop. And one important thing to note is that a lot of times in any form of cancer, it is not the primary tumor or the original tumor that kills the patient. It's usually the spread of the disease. It's the metastasis. So I'll take a minute to tell you that for me, my research interest is understanding the metastatic spread of breast cancer, particularly in triple negative breast cancer, because we can save a lot of lives if we know what's happening in the cell that's going to make it go and spread to your lung, your liver, your brain, or your bone. So I'm going to tell you a little bit about what I'm doing with my research to understand how triple negative breast cancer spreads. So now we want to talk a little bit about some of what we've learned from genomic sequencing about triple negative breast cancer. So don't be alarmed by the large data set that you're looking at on your screen. So key things you need to know. On this left panel right here, these are just different types of breast cancers. And then here, these are different genes that we think are potentially mutated in breast cancer. So basically, each of these genes, they encode for proteins that have special functions. But then if they get mutated, they might contribute to the development of cancer. So in triple negative breast cancers, which we also refer to as basal breast cancer at times, we understand very well that the most commonly mutated protein in triple negative breast cancers is TP53, or a gene called TP53. So in 80% of triple negative breast cancers where we have sequencing information, and where do we get this sequencing information? When patients come into the clinic and we are taking biopsy samples, or we are sequencing, or the tumor has been removed, and then maybe they started treatment, or we sequence their genome before they began treatment and after, so we can track response. So here is where we are collecting data from the patient side to understand, well, you know, we have this set of patients that come in with this type of cancer. We can sequence them and try to understand, is there something common that may be driving this cancer? And what we know well about patients whose genomes have been sequenced where the patients have triple negative breast cancer, we know and understand very well that the P53 gene is going to be mutated. So I want to tell you a little bit about P53, because that is at the heart of my current research at MD Anderson. So P53 is what we call the guardian of the genome. I'm thinking maybe someone may need to mute. Okay, don't hear the feedback now. So we call P53 the guardian of the genome, and we call it, can you guys hear me well?
Yeah, it looks like there was a phone. And I think they hung it up.
Okay.
Thank you. I was trying to mute it, but I couldn't mute it. And I was like, oh, goodness.
Okay.
All right, thank you.
That's okay. So we understand that P53, we call it the guardian of the genome. The reason why we call it that is because if you can remember when I showed you the picture of where your cells are making copies of one another, and there are protective measures set up in case you have a mutation or something is wrong with your DNA, there are triggers that tell the cell to kill itself. Well, the most important protein that tells everything in your cell to go and kill itself is P53. So this is a very busy slide. The only thing that you need to know is that P53 is a special protein that is activated when you have changes that affect or assault your DNA. So your DNA can get broken. You can have mutations. You can be exposed to some type of UV radiation that will cause your DNA to break. Or you can have other proteins that are being active that promote cancer and are telling your cells to keep growing, keep dividing. Whenever the cell recognizes any of these bad things happening, P53 is activated. And what P53 does, it's going to go and bind to your DNA, and it's going to say, hey, turn on some genes to make proteins that are going to cause cell death. It can also try to help the cell to fix itself or tell the cell to stop growing. But the key thing to know is that P53 is going to try to prevent the formation of cancer cells. So P53 really protects you from developing cancer. And the cool thing about it is that I think a lot of people fail to realize this. Our bodies see unhappy cells or cancer-like cells or cells that have the potential to be cancerous all the time. But because of this protein, when it's operating correctly, it tells those cells to go die and you don't get cancer. However, from sequencing different cancers, breast cancer, head and neck cancer, bladder cancer, what we have learned is that P53 is the most highly mutated gene in cancer. So key things you need to know here. We are just looking at a graph, and what it's doing is measuring the number of different tumor types. So on the bottom are all the different types of cancers that we do have sequencing data available for. And then telling us how often in these types of cancers do you see P53 mutated. And in the context of breast cancer, we see that P53 is mutated in about 40% of all breast cancers. Now, when P53 becomes mutated, it loses its ability to go and tell the DNA to flip on genes that are going to help protect your cells from forming a cancer. It loses that ability. So we know very well from sequencing data where the mutations in the P53 gene occurs. And the only thing that you really need to take away from this top part of the figure is that there are specific mutations in the gene of P53 that are going to give you a higher risk of developing cancer. We call these P53 hotspots. They're hot because if a person is going to get a P53 mutation and develop cancer, it is likely going to be at one of these specific points in the P53 gene. So not very important for you to know any more than the hotspot. You will see me use the term 248 or 245 interchangeably for the next few slides. The only thing you need to know about 248, that this particular hotspot is the most aggressive in cancers, particularly in breast cancer. A lot of people that have this particular mutation, they're likely going to get an aggressive form of cancer that is likely going to spread to the rest of their body. So I've told you a little bit about P53 and its role in trying to protect you. And I've told you that once it's mutated, now we're dealing with mutant P53. And mutant P53 is a whole different beast. In fact, mutant P53 is doing nothing to protect your cell from cancer. What it does now is telling your cells, hey, keep growing, keep dividing, go form a cancer. And it's a pretty smart protein because I've told you that mutant P53, it can no longer go to your DNA and say, hey, turn on protective genes. It's lost that ability to bind to your DNA. So what it does, it hijacks healthy proteins that still have the ability to bind to DNA and turn genes on, and it makes it do bad things. Some of the things that mutant P53 is going to tell your cell to do is to migrate. Basically tell the cells to move. Not only are they going to grow out of control, but they want to leave the tissue that they're in and they want to venture out to the rest of your body. So basically it's going to promote invading and metastasis so that we can set up shop in other organs of your body. So not only do you now have the original tumor, in the case of breast cancer, it's the first tumor, but now you're beginning to form tumors in other locations of your body. Other bad things that it tells the cell to do is to be resistant to chemotherapy. So whenever you hear patients say that they may be changing their chemotherapy because they were not responsive to the first one, there may be mutant P53 lurking in the background that is causing the cells to not respond. So basically, mutant P53 is going to provide some advantages to the cancer cells so that they can just keep going and growing and ignore any type of signals that are trying to get the cells to die. So at the heart of my research is trying to understand what is mutant P53 doing to drive breast cancer? Because if you remember, I told you that P53 is mutated a lot in triple negative breast cancers. So I've walked you all the way through understanding the genome, the technology we use to read the genetic sequence, and what we've learned from understanding the genome in the context of breast cancer. And that has let us know that P53 is potentially important in triple negative breast cancer because it's mutated there. But it's great to be able to understand the human data. Sequencing has revolutionized that. However, we cannot do research on patients. We have to be able to have a good model. So what I am doing, I am actually using a mouse to try to understand this. So the key things that I want you guys to know about using the mouse is that we understand the genome very well. And it's very close to the human genome. So it's a great system for understanding the biology of different types of cancers. So what I do every day, I work with mice, and mice have breast tissue, and I can go into the mouse and I can inject a construct of DNA that allows me to go from having normal P53, so the mouse is normal and healthy, to then when I do an injection, it turns on this mutated form of P53. And what happens when we switch on mutant P53? We're able to form breast tumors. So I know this is pretty gruesome, not what you want to look at on lunch, but I'm showing you this so that you can know that when we use animal models for cancer, a lot of times they will tell us some very true characteristics, very similar to human biology. And here we are able to get breast tumors in the mammary glands of the mice. And here, these are just images of what the tumors look like. And this is where we're just showing you a picture where I can track where I've injected the where I've switched on the mutation inside of the duct of the breast. So these are actually breast cells that I have lit up that now have this P53 mutation. The cool thing about switching on this P53 mutation is that we are getting triple negative tumors. So now we have a very good model for understanding triple negative breast cancer. And I'm very excited about it because now I can start to pinpoint what are some of the things that are allowing mutant P53 to make triple negative breast cancer so aggressive, and hopefully learn something that will inform therapy. One part of the project is to understand how breast cells leave the breast tissue, go into the bloodstream, and start setting up tumors in other organs. So understanding what's going on with these circulating tumor cells, that is a potential target for therapy. Why do we care about this? Because in the clinic, we're able to sequence the patients. We can understand whether a drug is working or not working. But eventually, you cannot keep going back to the same human getting tissues. But if we have very good models that reflect what we see in the patient biology, now we have unlimited access to tissues. We can test whether or not drugs work. We can learn more about the biology of different tumor types. And this is the heart of what we are doing every day on the research side at MD Anderson. So we're known for revolutionary patient care and treatment, but the research side is equally revolutionary because we are very good at modeling cancer. So I'm very excited about this. This is actually toward the end of my presentation, because one thing I didn't tell you is that with this mouse model, it takes 2 years to develop the tumors in the mice. So now, just before pre-quarantine, I just started getting breast tumors. So if I have the opportunity to speak to you guys, maybe 6 months to a year from now, I'll be able to share more of the exciting biology and data of what we're learning from sequencing the tumors of these mice. So that's all I have for now, and I will quickly acknowledge my lab. Here is my advisor, Dr. Gigi Lozano. Most people have a hard time pronouncing her name. We just call her Gigi. She is a great leader, and this is the rest of the group, and we are all working very hard to make cancer history. And I will take questions from you guys now.
Awesome. Thank you so much, Dr. McDaniel. We really appreciate it. A lot of great information. And we got a lot of questions that actually came through. So I'll try to go through them. Let me spread my screen out because there's a lot of questions. All right, so the first one, it says, does sequencing the genomics allow to determine mutation process as a preventative measure for cancer?
Yes. So
Okay.
one example of understanding that is that if we understand what mutations are present traditionally in different cancer types, now we can start monitoring a little bit more. So if you have your genome sequence for whatever reason, just for fun, because a lot of people do it just for fun too, if you by any chance happen to have any changes in your DNA that suggest, okay, well, you've got this particular mutation, then you might be more at risk for developing this type of cancer. This is very important when we think about a mutation that is found in all of your cells. So these are what we call germline mutations. So the people who have the hereditary cancers, we're going to find those types of mutations. So you may never be thinking about, oh, well, I'm going to develop cancer or this or that. But sometimes we find changes in your DNA that may give you a predisposition for developing cancer. And for the people who do have these particular mutations, preventive measures can be taken. The best idea of that is in breast cancer, where women have BRCA1 or 2 mutations, and then they start thinking about their options. Do they want to go ahead and have a mastectomy? Do they want to have an oophorectomy? And these are all personal choices. The doctor is going to give you the data. You get to think about it. You get to think about what you want to do next. And I will tell you, with sequencing your genome, we're going to find a lot of mutations, and a lot of them we just don't understand, and we don't know what to do about them. And that is where the beauty of understanding the mouse models comes into play, because just like we've built this mouse model for P53, we can take this same tactic and apply it to other genes. We can learn about other tissues where genes are affected. So patient participation in the science is critical. We're all in this together. It's not just the research. It's not just the clinicians. We're all working together to try to understand what's happening in your body and how can we prevent or effectively treat you.
Awesome. I know we got about 5 minutes left, and there are a lot of questions. And if you all have time to stay, then I'll try to get through all the questions. The next question is, if a parent had lung cancer, would that predispose the child to breast cancer?
Not necessarily. Lung cancer is not necessarily a direct predisposition for breast cancer. However, just having a relative that has had some type of cancer, it does predispose you just from developing other types of cancers. But there is not a direct correlation between having lung cancer and breast cancer.
Okay, the next question, is there a need to repeat genotype test post-treatment?
Actually, you can do that. The reason why you would want to do post genotyping after treatment is so you can see if there are any changes to your DNA. Now, in the case of where the patient is very responsive, yes, we're going to take a post genotype because we want to know what changed in your DNA that made you respond to the treatment. So as you're going through treatment, your DNA is responding to that treatment. So either it's going to respond in a good way or a bad way. So either way, if you respond or don't respond to treatment, yes, we want to know what's going on with your DNA. So yes.
Is there any research that's been done to show the difference in treatment for African American versus whites? And the next question follow-up is, how does genomic testing reflect those differences?
Well, so let me tell you, backtrack a little bit. So what we understand well about genetic predisposition for breast cancer, these mutations are traditionally found in women that have Ashkenazi Jewish descent. So this is where we first identified this mutation in the Jewish population. So you have representation from one race a lot in a lot of these genetic studies. I will say that African American women are highly underrepresented in any of these types of studies, whether it's the clinical trials or where people are taking sequencing data from patients. We're just not there. But I think the key to that is having sessions like these where we are educating the public. And one part of that dichotomy is just because in the African American community, let's be real, we really have like a strong distrust of studies. When people talk about that, we tend to stay away. However, we have to get properly educated on why some of these things we should participate in so that we can better understand. Because if we don't have representation in these sequencing studies, we may be missing a lot of information as to why African American women are disproportionately affected by certain types of breast cancer. And there is a researcher, well, she's a clinician in Michigan, and she actually inspired me to even think about triple negative breast cancer. I had never heard of her, but she sees a lot of Black women that have triple negative breast cancer. She was highlighted on CNN like 10 years ago, and I was like, I've never heard of this, but I want to study that type of cancer. She's taken it a step further where she's trying to connect it to the genetics of women in Nigeria, because the majority of Black women that are here in America, we're descendants of the Ivory Coast. That is where our people came from. So there is likely a genetic predisposition that is in women of African descent as to why we get more aggressive forms of breast cancer as well.
All right, the next question is, sorry if I missed this, maybe I did. So triple negative cancers are rare in general population, but more common in young African-American women?
So triple negative, yes. When you consider all the different types of breast cancer, it is not as common as the hormone receptor positive breast cancers. So, and yes, we do see higher diagnoses of triple negative in African American women. It sounds like a dichotomy because we also are a minority just in the general population. But that's when you also have to think about like environmental factors. Like I said, your DNA is a function of your environment. We didn't really dive deep into how your DNA responds to your environment. We know that we have women that oftentimes do not have access to the best resources in patient care. So even if there is something wrong, they may not be going to the doctor. So they may have a cancer, don't know it until they're sick. Another option is also the diet. Some of the things that we eat. Taking care of yourself is very important. Eating well, being physically fit. And sometimes in certain instances, Black women don't have the time to do these things because we're dealing with other things in the world. So this is also there's a social component to it too. And there's a researcher at the University of Chicago, and this is the heart of her research because she is trying to understand why she sees more triple negative breast cancer in Black women in Chicago. And if you understand what's going on in Chicago, you understand why she wants to study how the environment affects certain women and why they get certain types of breast cancer. So your genome is very complex. It's not just you and your genetic makeup, is everything that you're doing. So we should try to live like stress-free lives. Sometimes it's hard, but we have to make sure that we're being healthy and taking care of ourselves too, because having constant stress, it just pushes your genome toward a way of being in an unhealthy state.
What are the results of Xeloda on TNBC in your research?
Well, I actually have not tested any type of therapeutic targets in my research per se, so I cannot speak to that exactly. So that's one of the goals that we're wanting to do with this mouse model, because we do think that it is a very good preclinical model for breast cancer and understanding what are potentially effective therapies. So instead of just testing it on a patient, we can test it in the mouse. Do we get rid of tumors? If so, then the next step is to try to push it toward a clinical trial. So I can't speak to that at the moment because we're in the very early stages of this model right now.
Okay. Dr. Joy, I know we've hit our time. We're now at 1:33 and we have a lot more questions to go. I don't know if you have time to answer any additional questions or if it's something that we can email you and maybe you can respond to it at a later time. So I'll leave that to you and you let me know if you want me to continue on or if you don't have the time for us to continue through the questions.
Actually, if you guys can email me the
Okay.
questions, I will
Perfect.
be more than happy to respond because I'm actually scheduled to do another Zoom call as soon as this is over,
Okay.
speaking to some grad students about careers in science. So I didn't
awesome.
realize I double booked today, but I am very happy I had a chance to spend time with you all. I've enjoyed the questions. I wish we could have a little bit more discussion if you guys even want to have a follow-up because we're all in quarantine now. I am happy to do that as well because I like talking to the people. So whatever you guys want to do, however you want to proceed, I'm happy to do it. But in the meantime, you are more than welcome to email me. Miss Loretta, you guys can share my email with everyone that is on the call and I'll get back to everyone.
Awesome. Thank you so much. We really appreciate your time spending with us. Like I said, this topic is so important, especially when we're talking about triple negative breast cancer, and to educate us as patients, you know, our goal is to empower patients to take control of their own health and being the CEO of their own health. So a lot of the information you shared gave us a great insight. So thank you so much, Dr. McDaniel. Loretta, you want to close us out? Is there any parting words you have?
Well, I'm just so glad that so many joined us. This is a nice group. I was trying to get to her email so I can put it in the chat box, unless you want to do it a different way.
Okay, well, we can send it out. We can send it out to everyone.
Okay, we'll do that then. We'll just send it out to everyone. But yes, thank you so much. Great job, Dr. Joy. Thank you so much for taking the time.
Thank you. Stay safe.
Thanks, everyone.
All right. Bye-bye.
Bye-bye.
Well,
okay, Miss Loretta, you're now the host.
Okay.
So you can end the recording.
Okay, you want me to end the recording?
Yeah. Thank you.
Oh, where it is? Oh, it's up there.
Yeah, it's at the top.
Okay.
I kept trying to click the button. I was like, oh, shoot, I don't have the rights no more.
Oh, I didn't know you had given it to me. Okay.
Well, I had given it to Dr. Joy.